progress.
Hello.
Hello.
Hello, how are you? Dr. Sylvie?
I'm fine.
Ah,
And
good,
you?
good. Yes,
Dr.
good.
Wibowo.
Yeah. Good,
Happy
good.
to see you.
Happy to see you too. Yeah.
And I'm very happy to be with you this afternoon for you, morning for me.
Yes, 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 yes. Okay, just waiting maybe for 11 minutes left. Okay.
Okay, let me share my screen.
Okay, okay. Please try. Yes, perfect.
Okay, so what do you see? Do you see the,
This is small. Yeah.
okay, let me check.
Can
that.
you
Do you have it in full screen or not?
not yet.
Okay, let me do something.
Okay. Yeah, that's perfect. Yeah, this is
Okay,
no full screen. Yep. Okay. Please wait maybe for 10 minutes left.
sure.
Yeah. Okay. Ya, kita menunggu 10 menit lagi ya untuk kita mulai. Ya, terima kasih. Good afternoon, everyone, ladies and gentlemen. First of all, I would like to thank you very much for Professor Sylvie Manguin for being willing to give a lecture for us. Very happy to see you again. And then, esteemed faculty member and student of Master Program of Tropical Medicine Gadjah Mada University, and the distinguished participant, especially from the Ministry of Health of Indonesia, and then National Research and Innovation Agency, and other research institute in Indonesia, Indonesian Pest Control Association, university members, and all of the participants that I cannot mention one by one. So welcome to today's guest lecture organized by Master Program of Tropical Medicine, Faculty of Medicine, Public Health and Nursing, Gadjah Mada University. I'm delighted to introduce our distinguished speaker, Professor Sylvie Manguin, who will enlighten us on the topic of the importance of mosquito biology and taxonomy in vector-borne disease control. Professor Sylvie Manguin is a full research professor at the Institute of Research for Development, IRD, based at the University of Montpellier in France. She is a leading medical entomologist and academician researcher whose main interests concern mosquito and vector-borne diseases such as malaria, dengue, and Japanese encephalitis. She has developed studies on the Anopheles mosquitoes from 3 continents, Asia, Africa, and also Americas, including molecular species identification, population genetics, phylogenetics, vectorial capacities, spatial surveillance, mid-gut microbiota biodiversity, salivary immunological markers, and vector control approaches. She is the author of more than 90 indexed publications, 6 book chapters, 3 books including Anopheles mosquitoes, New Insight into Malaria Vector in 2018, for which Professor Sylvie is the editor in Text Open Access, and the other is Biodiversity of Malaria in the World in 2008. She is also the Secretary General of the International Federation of the Tropical Medicine and member of the editorial boards of the Malaria Journal and Acta Tropica, and she serves as reviewer in several international institutions and more than 20 scientific journals. So please join me in a warmly welcoming Professor Sylvie Manguin as she takes the stage to share her presentation. So Professor Sylvie, the floor is yours. Thank you.
Thank you very much, Dr. Triwibowo Mbargajito. That's such a pleasure to be with you all and to see you, Triwibowo, because we had a long collaboration already and a very successful one, and I'm very proud of it.
Thank you very much.
I would like to speak about mosquito biology, also taxonomy and vector-borne diseases, and for the goal of the control of these vector-borne diseases. First of all, I would like to give you a few definitions about vector-borne diseases. What are they? One of the major cause of mortality in the world, if we count the mortality due to malaria and dengue. Vector-borne diseases are also an obstacle to socio-economic development in southern countries. And as you know, it's a very variable situation depending on the country. So now, what is a vector? So here I'm going to speak about insect vectors that transmit a pathogenic agent to humans or animals when they bite. The definition of a vector in this case is that the development cycle and the reproduction of the pathogen, whatever the pathogen, parasite, bacteria, or virus, has to be done in the arthropod. So the multiplication of the pathogen will occur in the insect. And the pathogen cycle necessarily passes through a vector and a host. So there is no host to host cycle. It has to go through the vector. So that's the definition of an insect vector. And as you know, there are a large number of insects that are capable of transmitting pathogens. So we all know about fleas, vector of the Bacillus Yersinia pestis, responsible for the plague, kissing bugs, also called triatoms, that are responsible for American trypanosomiasis or Chagas disease in South America. We have also tsetse fly or Glossina, which is the vector of parasites of sleeping sickness in Africa. We have also black flies, Simulium, responsible for the transmission of onchocerciasis, and sand flies responsible for leishmaniasis. But above all these different insects, the mosquitoes are the #1 vector of human pathogens. If we speak about malaria, lymphatic filariasis, dengue, and other arboviruses. So what makes a vector? So there are different factors, because not all the mosquitoes are vectors. So one vector has to be susceptible to infection. It's what we call vector competence. The contact with its host will suffer from the disease. So we are talking about humans or animals in terms of the hosts. The longevity of the insect, and we are talking about mosquitoes here, has to be long enough to allow the cycle of the pathogen into the mosquito before its transmission. And the abundance is also a factor that can be, not always, but can be when the vectors are in large densities, it will transmit the pathogen more easily to more people. So the vector that can cause numerous diseases in humans. And the numbers or the percentage show that 80% of the world population is at risk of one or more of these vector-borne diseases. So it's really a huge percentage of the population worldwide that is at risk. And not only in tropical countries, but also in temperate countries. The burden, we are talking about 17% of the global burden of communicable diseases are due to vector-borne diseases. And in terms of the mortality, it's an estimation, of course, but it's about 700,000 deaths due to vector-borne diseases per year. So it's, again, a huge number. So I'm going to speak now about the biology and how to recognize a mosquito, and obviously the genus and the species. So I'm going to, we are within the class of insect here, and you can see that in the insect there are many different orders. I am within the Diptera. Diptera means 2 wings. And within the Diptera order, I will be talking today about the Culicidae, which are mosquitoes. And these are the disease that they can transmit. So you can see that within the insect, there is a large diversity of other vectors. So within the mosquitoes, you have to know that 10% of them are vectors of pathogens responsible for the different diseases that we will be talking about. That means that 90% of the mosquitoes are not vectors. So it's very important to differentiate the vectors and the non-vectors. If we apply a vector control, we have to apply it to target the right vector and not non-vectors. The female who feed on blood can carry a disease agent from one human to another as they feed. So we are targeting the females also. And among the mosquitoes, there are about 3,500 species, and with three major genera of mosquitoes. And here we are going to speak about these three genera, which are Anopheles mosquitoes that transmit mainly parasites and mainly Plasmodium responsible for malaria, but not only. Anopheles can transmit also few viruses. And we are going to speak about Aedes and Culex that are transmitting viruses, but not only, they can also transmit parasites. So this is the mainstream I would like to show you, but knowing that there are exceptions. And the mosquitoes occur in all types of environments. We can find them in tropical, temperate, Arctic climates, but also from sea level up to 6,000 meters. And you have to know that the Anopheles mosquitoes can transmit, for example, malaria in very high altitude, up to 4,000 meters. So it's not because we are in high altitude that we can be safe from malaria. In some areas, the mosquitoes can hide and still transmit malaria. And we can find also them in caves and mines, so they hide in these places also. So it's very difficult to control them in some places. So here are the main genera of mosquitoes I'm going to speak about today. We have here the Aedes aegypti. I could show also Aedes albopictus, the tiger mosquito. They are both vectors of viruses such as dengue, chikungunya, Zika, yellow fever, and many others. Here we have a photo of Anopheles albuminus. But knowing that there are a good number of Anopheles species that are vectors of parasites, including malaria agent. And here we have Culex quinquefasciatus that can be a vector of arboviruses, such as encephalitis, a virus of the different encephalitis, such as Japanese encephalitis, St. Louis encephalitis, etc. So I'm going to show you the mosquito cycle. I'm not quite sure at what level of knowledge you are in terms of mosquitoes, but I would like to go through the basics so you understand a little bit more about mosquitoes. So here we have the eggs of a mosquito. From the eggs, we will have the larvae. And we have 4 stages of larvae. So we have the L1 that will give the L2, L3 to L4. The L4 will give the pupa. And from the pupa, there will be the metamorphosis into the adult. And here we have an adult emerging from the pupa here that you can see. Another pupa on the right side is still with the adult inside. So the adult will emerge. It will be either a male or a female. We can see here that it's a male, but I will show you later how to recognize them. And mosquito mating will occur, and then the female will search for a host to bite, to get blood. And every 2 days, the female will bite, lay eggs, bite, lay eggs. So that's the life of a female mosquito. So you can see that there is an aerial phase for the adults and aquatic phase for the immature stages. I would like to show you the different larval habitats of these 3 genera of mosquitoes. So here we have different larval habitats for Anopheles mosquitoes. Usually they are very natural areas, so a stream, a pond, stagnant water, wherever this is in Africa. Here, these 3. Here it's in Vietnam. You can find them also in artificial containers, although rare, but it occurs. So for Anopheles mosquitoes, you have to understand that the water is rarely polluted, although it can be very turbid, like here, because people are digging to make bricks, but the water is not polluted. So this, although there are some exceptions, but in general, it's like that, natural water. For the Aedes habitats, you can find Aedes in stagnant water in pretty much every container that has some water. So it can be a tree, a hole, gutters, whatever that plants with a cup under with stagnant water. You have Aedes mosquitoes, and for that, the Aedes mosquitoes are closely linked to humans. And these 2 that with a circle are the way the tiger mosquitoes travel to the different continents. So you know that the tiger mosquito is coming from Southeast Asia, and through the boats with tires or lucky bamboos that has water at the bottom in a plastic container. They travel like that. And the mosquitoes were or are in the tires or in these lucky bamboos, and they came into the different continents, and now we have tiger mosquitoes everywhere, in Latin America, in Africa, in Europe, everywhere. So now for the culex, on the opposite of the Anopheles larval habitat, the culex, they really enjoy polluted water. So you find them in dirty water, pretty much, closely linked to cities and artificial habitats, so urban, on the contrary to Anopheles that is more in rural areas. So from eggs to adults, the female can lay between 50 and 300 eggs every 2 days at the surface of the water. The larvae will hatch in the water and undergo 4 stages, as I said, coming from the small L1 to the big L4, changing size each time, interspersed with molting, and it lasts about 4 to 10 days, the cycle of the larvae. The larvae feed on plant debris, on algae, and the 4th instar larva will change into the pupa. And the adult will emerge within 2 or 3 days after transformation or metamorphosis into the pupa. And the female adult will live about a month, between 3 and 4 weeks. Of course, as you may know, the female needs blood meal for the maturation of the eggs. So here you have you can see the mating of the mosquitoes. In fact, it occurs for most mosquitoes. You have a swarm forming during the dust time with few males, up to 1,000 or more males together, forming this swarm with a big noise. And the female will be attracted by this noise, will come close to the swarm. And when the males see or hear the females coming, they leave the swarm and they follow the female for mating. And the female has an spermatheca, which is a small organ in which she will keep the spermatozoides. So usually the mosquito female will mate once, and she will keep the spermatozoids for the rest of her life. So the mosquito eggs are different if we are talking about Anopheles, Culex, or Aedes. So you see here Anopheles egg with a floater. Here you have the eggs of Aedes without floater, and but with the very interesting characteristic is that Aedes eggs resist to desiccation, so they can dry, no problem. And at the next rain, they will the larvae will get out from the eggs. So the eggs can last for years without water, and then you put some water and the cycle starts again. The Culex egg, the Culex female will lay eggs in a raft like here. So these eggs are vertical and they form this raft. So you can see here the size of the larvae from L1. This is an Anopheles. These are Anopheles larvae. So the L1, and then after molting, we will have the L2, L3, and you can see the big size of the L4, and here the pupa. And how to recognize Anopheles from Aedes and Culex? It's that Aedes and Culex, they have a siphon to breathe, whether the Anopheles larvae doesn't have a siphon. So you can see here, without a siphon, the larva of the Anopheles will be parallel to the surface of the water. Whether for Culex and Aedes, with the siphon, you can see that the position of the larvae is either vertical or on the sides, but not parallel to the surface. The mosquito wings are very important because you can see that in the case of Culex or Aedes, there is no, the scales on the wings are very rare. Whether for Anopheles, the Anopheles wings, they have many scales and dark or white, and these scales will be very important morphological identification characters. So the wings are very important to identify the Anopheles mosquitoes. To differentiate Culicinae from an Anopheles, you can see the scutellum. So here you have the head, the thorax, and the scutellum is has 3 lobes for the Culicinae, that means Culex or Aedes, and rounded scutellum for the Anopheles mosquitoes. For the Aedes thorax, the identification is not so complicated for the Aedes. You can see Aedes aegypti here. You have this S shape on the thorax for Aedes aegypti, whether for Aedes albopictus, you have the white line in the middle of the thorax here. So very easy to recognize naked eyes, which is not the case for Anopheles mosquitoes, but we are going to speak about that a little bit later. So for the abdomen and the legs of Aedes, why we call the Aedes albopictus a tiger mosquito? Because as you can see, there are black and white bands on the thorax, on the abdomen, on the legs. So that's why we call it tiger mosquito. Now, the resting position also is different when we are talking about an Anopheles or Aedes culex female on the surface. So the Aedes, the Anopheles mosquitoes are oblique from the surface, as you can see here. Whether Aedes and culex, the position is parallel to the surface. So that's another way to differentiate Anopheles from the two others. Another very important aspect that you have to know is that the female Anopheles and culex bite at night from 6 p.m. to 6 a.m., more or less. But they bite at night, and Aedes mosquitoes, they are day biters. So yeah, it's difficult to escape from a mosquito bite because during the day, the Aedes will bite you, and during the night, Anopheles and culex will bite you. So very difficult to escape. So the morphology of an adult mosquito is that you have here the head, the eyes, the head, and the proboscis, the antenna, the palps. Here you have the thorax. On the thorax, you have the 3 pair of legs and the 2 wings. I said they are dipteras because they have 2 wings. And under the wings, there is the abdomen. So on an Anopheles mosquito, but for the other mosquitoes as well, all the different patterns of scales, the shape of the legs, the head, the proboscis, the length of the proboscis, these are morphological characters that can differentiate at least the genera and sometimes the species. So these mosquitoes are hematophagous mosquitoes, especially as I said, the female. The males are not. The males are plant nectar feeders, as well as the female for the energy. So only female will bite for egg maturation, not for feeding themselves. Not all mosquitoes bite humans. And as I said, 10% of the mosquitoes are vectors of pathogens. And here you can see that the female possesses 6 perforating stillets. So you can see here the very sharp stillet that will penetrate the skin to find the capillary and take the blood meal. And the labium, you can see here the labium is the protection of the different stillets. So when the mosquito female is biting, the labium is putting aside, and when the female is done with yeah, you see the labium here also. When the female is done with the biting, all the stillets will get into this protection that is the labium. So it's very performant, very well done morphology. If we cut the proboscis of a mosquito, you see the different stillet and which are mandibles, hypopharynx, etc. I'm not going to speak about that. What I want you to see is that a mosquito has 2 channels or duct. One is the food channel in which the blood will get into the mosquito. And here you have the salivary duct in which the salivary, including the parasite when the female is infected, will go into the host. So it's like one is going in, the blood is going in, and the salivary and the parasites or the virus are going out. So the female injects an anticoagulant with the within the salivary duct to prevent clot formation, and that will generate this irritation reaction that we all know. The salivary containing this anticoagulant is injected through the salivary duct, and the pathogens are also transmitted via the saliva. So how to recognize a male from a female mosquito? The female we call the antenna hairless. So you can see here the antenna hairless, although there are few hairs, but not so much compared to a male. You can see here a male. So we call the male with a fair or feathery antennas. So now we went through the differences between Culicinae and Anopheles. So here's the synthesis of the differences. So Aedes debiter, oops, sorry, mosquito, Anopheles culex night biter. And you can see here the Anopheles with the disposition, the wing, the eggs, the position of the larva and the pupa, compared to culex and Aedes with the different position, different wing, different eggs, position of the larva also different. The pupae are very difficult to distinguish, but so you have the recapitulation of the different characters here. So now speaking about vector-borne diseases and vector identification. I'm going to speak about malaria because, and start with malaria, because this is the main problem for species, mosquito species identification. So I will spend a little bit more time on malaria than on the other diseases. So here you can see an Anopheles mosquito. The children under 5 years old are really pay a very heavy price, because between 0 and 5, it's where these children are quite vulnerable, especially in sub-Sahara Africa. And here you can see the parasite Plasmodium into the red blood cells. And here in green, you have the sporozoite coming from the mosquito. So the sporozoites are the infectious stage of the Plasmodium. This is a map that I've done a few years ago, but I updated it. And you can see that malaria, as we all know, is in the tropical belt, sorry. But what I want to show here is that it's not homogeneous. It's not the whole tropical belt that has malaria. There are very much differences. Like in Latin America, it's mainly Plasmodium vivax that is occurring. In Africa, it's Plasmodium falciparum. Now in Southeast Asia, it's for many countries, mainly Plasmodium vivax and Plasmodium falciparum. In Papua New Guinea, Plasmodium falciparum, and we are talking about the very high prevalence of Plasmodium falciparum in Papua New Guinea, Solomon Islands, such as Africa. Whether in the rest of Indonesia, we have less falciparum and more vivax and the other Plasmodium parasites. So you can see what I want to show here is the heterogeneity of malaria throughout the world. So the cycle of Plasmodium, there are 5 species of Plasmodium transmitted to humans. The main ones are Plasmodium falciparum, the deadly Plasmodium, but also Plasmodium vivax, malaria and ovale, and more recently Plasmodium knowlesi, which is a simian malaria parasite that now occur in humans. So as you can see, the vector, the Anopheles mosquitoes are the vectors, and the cycle is occurring in the mosquito and as well as in the human host. I'm not going through the different stages because I'm sure you know the cycle quite well. In the mosquito, it's very interesting to see that the mosquito female will get from the infected humans the gametocytes. So the gametocytes will get into the midgut of the mosquito, and the gametocytes will give gamete males and females. So you see a male here, female here, and they will give a zygote. This zygote will cross the midgut membrane, and to get to become an oocyst outside the midgut, in which the parasite will multiplicate, and within when the oocyst is mature, the oocyst will open up and give sporozoite, as you can see here. Sporozoite being the infectious stage, and this sporozoite will move into the salivary glands here. So this takes about between 8 and 20 days, the whole cycle here, depending on the mosquito, depending on the temperature, depending on different factors. But the average is between 10 and 12 days, but it can occur also in 1 week. And when the female has the sporozoite in her salivary glands, the female will be infectant all her life. So every 2 days, she can infect a new person until the female will die. So this is very important to understand this cycle. Within the Anopheles species, there are about more than 530 species of Anopheles, but only 65 to 70 are vectors of malaria agent of Plasmodium, and 41 species are considered dominant vectors. So they can transmit 5 species of human Plasmodium. I just gave you the names. And today, the malaria is a major burden of human health. I mean, today, for centuries, has been a major burden of human health. And in 2022, the WHO estimated that in 1 year, this year, there has been 608,000 deaths due to malaria. So I showed you the map of malaria in the world. Now I show you the map of Anopheles, and especially the dominant malaria vectors in the world. So you can see that this map is also very heterogeneous. But what you have to remember from this map is that each continent has its own dominant malaria vector. So in America, you can see the species are not the same in Europe, not the same in Africa, etc. So each continent has its own malaria vectors. And for example, the dominant malaria vector in Latin America is Anopheles darlingi. In Africa, it's Anopheles gambier. In India, it's Anopheles culicifaciens, Anopheles stephensi. Of course, there are others, but these are the main ones. In Southeast Asia, we have Anopheles diris and minimus. And in Indonesia, Malaysia, we have Anopheles leucosphaerus, which is a complex, and Anopheles sandaicus complex. So these to give you some names, but it's more complicated than that, of course. And Anopheles faroti for the Papua New Guinea and the Pacific area. So to resume here, Anopheles darlingi is the main malaria vector in Latin America. In Africa, it's Anopheles gambier. And in Southeast Asia or in Asia, we have different species, Anopheles diris, Anopheles minimus, maculatus, faroti, and many others. So now we get into the complexity of Anopheles mosquitoes. And I want to speak today about this notion of species complex. So the Anopheles species complex, I want to give you the definition so you understand why we do all these different techniques, molecular techniques to identify the species. And that will be after this definition. So a complex of sibling species of Anopheles is composed of morphologically indistinguishable species. So you go to the field, you collect mosquitoes, they all look alike morphologically, but you may have vectors and non-vectors or secondary vectors. So you have to know if you want to implement vector control, you have to know if it's a main vector or a non-vector in order to target the right vector and not to spend money for a non-vector. So simple but robust molecular techniques have been developed and are very, oh, I'm sorry, why it's moving like that? So these molecular techniques must be applied to reliably identify Anopheles mosquitoes to the species level. Because with the morphology, we will go to the genus level, but you need to know the species to target the right species for the vector control. That's why you need to use molecular techniques. I will also speak about Anopheles group of, and this group of Anopheles species is composed of morphologically distinguishable. So some species in a group, you can separate them using morphological characters. However, some species or some stages cannot be morphologically identified to a species level. So the group is a mix of, yeah, you can use morphological characters, but for some species, you still need to apply a molecular technique. And here I want to show you the high diversity of Anopheles mosquitoes in Asia. So 61% of the Anopheles species belong to a complex. That means that for minimus, complex, Barbirostris, diris, leucosphaerus, sandaicus faroti, these are species complex. You cannot identify them using the morphology. That's very important to understand. You have to use molecular techniques to identify to species level. And that's really the case of Asia, where you have 61% of the species complex. You have also some complex in Americas, in Africa, in Europe. But in Asia, the situation is quite complex. So, as I said, in Asia Pacific, the morphological characters alone are not sufficient. You need to use the molecular techniques to identify the correct species. So when we have a close-up on Southeast, on Asia, you can see that there are 19 dominant vector species. And you can see from the colors that it's quite diverse. This is the paper by Sinka et al. And when we look at Indonesia, this is a very good example of this biodiversity of Anopheles malaria vectors, where you have a large number of species occurring in the western part of Indonesia. And when you look at Papua New Guinea, you can see that they have very they have their own species in Papua New Guinea compared to the western part of Indonesia. So they are in red here. So Indonesia, I would say that the Anopheles mosquitoes are complex and very diverse in Asia, but in Indonesia, it's even worse because you have a extremely large biodiversity, and it's where you really have to study the species and use the molecular techniques when they are available, or you have to develop techniques to identify them in the future. So you have a lot of work to do in Indonesia, and Dr. Triwibowo Ambargashito knows a lot about that. So now I'm going to show you the different molecular techniques depending on the complex. So for the Anopheles dirus complex, which is the most efficient vector in Southeast Asia, which is found in deep forest, heavily shaded forest. So you can see here a case in Cambodia and in Vietnam. Very anthropophilic. That means that this mosquito target humans outside. So it can get in the bite, it can bite inside the house, but very quickly the mosquito will go outside, and sometimes it bites also outside. So very difficult to control because the bed nets will not affect this mosquito. This mosquito will bite when people work in the forest at night or early morning or before going to bed. The habitats of the larvae are in shady, leafy, temporary pools, puddles, in dense forest, stagnant water, and sometimes artificial containers like here in Vietnam, but rare. And there are 8 species within the complex, from highly efficient vector to non-vectors. So this is the distribution of the Anopheles dirus complex in Southeast Asia. And here, this is the map of the 8 species within the complex. So the main vector is Anopheles dirus and Anopheles baimai. So you can see the distribution is different. One of the in the west baimai and one on the east for dirus. There are some local vectors like Anopheles crescens, which is also a vector of Plasmodium knowlesi in green here. Anopheles elegans in India, we have no data about being a vector, a local vector or not. And we have non-vectors such as Anopheles nemophilus in orange here, or Tagasagoensis, and the new species here in Vietnam, which is a non-vector also. This is the technique that my current Thai student has developed, and he published this article this year on a new PCR that can differentiate, as you can see here, 5 species within the complex of dirus. This technique is more efficient than the previous one the previous PCR developed by Cathy Walton in 1999. So because this technique by Cathy Walton was very difficult to implement, but this one is more reliable and quite efficient. So this is the new technique. For the leucovirus complex in green here, you can see that both complex dirus and leucovirus are overlapping over the Thai Malaysia Peninsula. And they are very closely related. So the dirus complex has 8 species, as I said, and the leucovirus complex 4 species. And the two complexes are very closely related. So there is a very high risk of misidentification among the species of both complexes. Especially, of course, in this area where they overlap, obviously. This is the distribution, geographic distribution of the leucovirus complex. So especially in Thailand, southern Thailand, Malaysia, and Indonesia. And here you can see the species, the distribution of the species in Sumatra, and Borneo, so Kalimantan, with Anopheles latens, balabacensis, leucospherus here, and on the peninsula, both latens and entrolatus. So entrolatus being the vector Plasmodium knowlesi, the simian malaria parasite. And here the recent study by Pramasivan in 2022 showed the distribution in Malaysia of the different species, including Anopheles crescens from the dirus complex here in orange. So you can see that we have balabacensis here, Anopheles latens in Sarawak, and on the peninsula, a mix of species as well. So that's the technique, the PCR technique that has been developed by Pramasivan in 2022 that can differentiate entrolatus from Anopheles crescens from the dirus complex and from 2 forms of Anopheles latens and balabacensis. So the technique exists to differentiate these different species. And that this I suggest that this technique is being used in Indonesia as well. So now for the maculatus group, this is also forest mosquito with which is, I mean, it's a group. So remember the group, there are some species that can be differentiated by morphology, but some others you need to have the molecular technique. And we find them in dense forest in the different pools. And this one has 10 species. So included in the group, you have 2 subgroups, the maculatus subgroup and the Sourdough-Porny subgroup, and a complex, the maculatus complex, with the Javanese form that has been found in Indonesia. So that's the geographic distribution of the maculatus group. And that's the map I have made with the different species that you can find. So in pink, the large distribution of Anopheles maculatus. In the Philippines, you have specific species of Anopheles dispar and greeny, and some species in Asia that are Pseudo-Wilmori, Sourdough-Porny, which are vectors of Plasmodium. So this is the publication made by Dr. Gargito on the Javanese, on the Anopheles maculatus in Indonesia, and with the new new species in Central Java. So the Javanese form, you have the wing here with the different scale pattern, and you can see that also there are different patterns in other maculatus populations. So what I want to say here is that the wing pattern is really unreliable character for species identification. So the Cathy Walton in 2007 and collaborators developed a technique which works quite well to identify or separate the different 5 different species, not the 10, but at least 5. And that's a gel that has been done in my laboratory with where you can see here some specimens of Maculatus, Pseudo-Wilmori, etc. And these are the controls of each species. So very easy technique to implement. When we compared the morphology of from the field of some of the specimens that we collected in the field, and then these specimens were identified, and then we applied the molecular technique on these specimens from the field, we found a very high, sometimes rate of misidentification. So for the minimus complex, 7.3% of the specimens were misidentified. For the maculatus group, 41.5% of the specimens were misidentified. For the dirus complex, 63.2% were misidentified. So you can see very high level of misidentification if we rely only on the morphology. And all the specimens we collected in Thailand, we thought they were Leucosphaerus complex. They were all misidentified. In fact, they were belonging to the dirus complex. So molecular techniques are mandatory for the reliable identification of species within complex and group. Now I would like to speak about the Barbirostris complex. There are 6 species that you can see here. Vanderwulpi is the species that is occurring in Indonesia. The others are being found in Thailand. These are also forest mosquitoes. The larvae are being found in the swamp, deep fresh water, still of slow moving, with sometimes some pollution. And this is the work we have done in Thailand, where mosquitoes have been collected pretty much all over Thailand. And we applied the PCR that has been developed in my laboratory. And we were able to see that Anopheles veischuchotai was found pretty much everywhere in red. The blue mainly in central and northern Thailand, whether in the south, Anopheles dissidents was occurring mainly in the south and in the eastern southern part of Thailand. So you can we did another study with even more specimens here. You can see here. And really that will be very interesting to do this kind of study in Indonesia. And I will not be surprised that you find new species. So this is the PCR technique that has been developed in my laboratory in 2019. It's a double PCR that we developed. So one PCR to identify the 5 here 4 different species. The 5th one is the one in Indonesia, but we didn't have specimens, unfortunately, at that time, but we developed the primers to identify the Anopheles van der Woolpie. So as soon as we have few specimens from Indonesia, we will test them with the technique. The second PCR is only to confirm or to double confirm that we have these specimens of Velschushotai for lane A or Anopheles sangue for lane C. But this is also very easy and reliable technique to separate the Barbirostris complex species. For the Sundaicus, you know, Sundaicus is a very important vector in Indonesia, found in brackish water, especially in shrimp farm, and it's a complex of 4 sibling species. So Anopheles epiroticus, Sundaicus sensu stricto, and Sundaicus species D and E now we know. And I'm going to show you the map. So here are some larval habitats, the photos I took in southern Vietnam. So you can see here on this map that it's really along the coast because it's mainly a brackish water mosquito so all over Southeast Asia and in Indonesia as well. And we doing some molecular work, we found that Anopheles epiroticus is found on the continent. In Anopheles Sundaicus species D is found in India, Andaman and Nicobar Island. Species E is found in Indonesia. And the true or the sensu stricto Anopheles sundaicus is found in Borneo, so Sarawak, Sabah, Kalimantan. Then we developed this PCR technique, a little bit complicated to develop, but easy to use. So you can see here the band for Anopheles epiroticus. Here the band for Anopheles sundaicus sensu stricto, and here the Sundaicus species E from Indonesia. We don't have species D from India because, as you know, the Indian don't want to share. Don't want to share. It's not allowed. It's not that they don't want, but it's not allowed for them to send specimens abroad. So that's why we don't have, we couldn't test Sundaicus D on this PCR. So this is reliable technique that has been used, and with the technique, a publication, a recent publication by Dr. Siafruddin in 2020, they found that there was Anopheles epiroticus also in Indonesia, in these 2 areas, northern Sumatra, and in this island of Bangka Belitung. I hope the pronunciation is correct. And so now the map is this one. We have an Anopheles epiroticus also in Indonesia, in these 2 areas. So it will be very important also for Sundaicus to do some more work to see if we have more sympatric species in Indonesia, maybe some Sundaicus being found in Sumatra, Java, or species E found in Borneo, Kalimantan. So we really need to check more in details what is occurring also for Sundaicus. For the Faroti complex, a lot of work has been done on this complex that you can find in Papua New Guinea, Solomon Island, in Asia Pacific area. There are 8 species that have been identified, and it's also a coastal mosquito. And here, this is the PCR that has been developed by Bibi and collaborators in 2013. So you can see that it's very efficient PCR. It's a RFLP that can identify 10 members of the Punctulatus group, including the different species of Faroti, Punctulatus, Coliensis, which are very efficient malaria vectors. So after showing you all these PCR different techniques that have been developed, I want to show you why it's so important to identify the species and not only the genus. So I would like to show you 4 examples of misidentification and some consequences of these misidentifications. So one is, it's a study that has been done in Vietnam, another one in Central Vietnam, third one in the Philippines, and the 4th one in India. So in Vietnam, they were targeting Anopheles, which they thought it was Anopheles minimus, which is a malaria, very important malaria vector. And so morphologically, it looked like Anopheles minimus. When they used the PCR that was developed later on, they found out that it was absolutely not an Anopheles minimus, but Anopheles varuna, which is not a malaria vector. So the molecular assay showed it was Anopheles varuna, a zoophilic species not involved in malaria transmission, which means that for years, the Vietnamese in this area were doing a vector control that was totally inefficient because they were targeting the mosquito that was not transmitting malaria. So think about the waste of money, waste of time for targeting a mosquito that was not involved in malaria transmission. So that's the first study of misidentification. Another case in central Vietnam, in which I was also involved, is the composition of malaria vectors in this area of Nha Trang in central Vietnam. So in this area, it was well known for years and years that the main vector of malaria was Anopheles minimus. And one day, the people working there contacted me and said, "Oh, Sylvie, this is strange because the mosquito we have now has a different behavior. It's not like before, it's biting more outside and we are not sure what it is." So they send me the mosquitoes, and in fact, what I found is that up to 1999, the mosquitoes were Anopheles minimus in blue. So you can see very high density of these mosquitoes, 100%, 97%, etc. And you can see in orange Anopheles harrisoni, which is sibling species of minimus, so morphologically identical, was very rare. And suddenly, in 1999, they implemented in the village impregnated bed nets. So they provided to the whole population bed nets. So the minimus that is usually an indoor mosquito, biting indoors and resting indoors, was in fact, killed, or the densities felt, and the mosquito that was not entering the houses was not affected by the bed nets. And this one suddenly, without the competition with the other species, exploded. Up to the point that in 2002, they found only Arisoni, 2% minimus, and 88% Anopheles arisoni. So this change is due to the implementation at a large scale in these villages of impregnated bed nets. So Anopheles minimus, more endophilic than Anopheles arisoni, was more in contact with insecticide, and the densities dropped. And on the contrary, Anopheles arisoni, more exophilic, was not entering the house, and the population exploded. So in this case, you can see that insecticide-treated nets or insecticide residual spraying indoor are less efficient against exophilic mosquitoes. So it was very difficult to control Anopheles arisoni. However, Anopheles arisoni is not as a good vector of malaria as Anopheles minimus is. So even though the density of arisoni increased, the malaria cases decreased because arisoni is called a secondary vector in Vietnam. Now the case of malaria vectors in the Philippines, using RFLP PCR, this technique indicated that some morphological Anopheles flavirostris, which is a main malaria vector in the Philippines, in this case also was misidentified, and it was in fact Anopheles philippinae, which is a non-vector of malaria. And this identification based on the morphology was using the dark scale area of the anal vein and the French coloration, which is unreliable character for separate these two species, Flavirostris for Philippinae. So this study also demonstrates the importance of using molecular techniques for reliable species identification. And the last case I want to show you is also misidentification between Anopheles minimus and Anopheles fluviatilis in India. So the molecular data indicated that Anopheles fluviatilis was not this species, but it was Anopheles minimus because of the climatic influence. And that's also an important aspect to keep in mind, is that the climate can change the color of the mosquito. And in this case, there are morphological variation within Anopheles minimus adult due to the cold climate during the immature stages. So the minimus looked darker than usual, and being darker, it looked like Anopheles fluviatilis. So you can see that also the climatic changes can also change the morphology or the scales or of the Anopheles mosquitoes. It's another reason why we have to rely on the molecular techniques. So the benefit of a correct species identification, it's that if you have the correct species identification, you will have an efficient vector control. If you don't know what mosquito you have, your vector control will be lousy, will not give you any efficiency. The second point is that most of Anopheles species, as I said, cannot be differentiated based on morphological character only. So if you cannot differentiate, you have to use PCR, and PCR assays have been developed now, as I showed you, for different species, different groups. So please use them when you go to the field, you collect your mosquitoes, you go back to the lab, you use PCR. If you publish something without this PCR identification, new paper, it doesn't worth anything. And if I get your paper without a PCR identification, your paper will be rejected. Be sure of that. So please, this is important. And as I said, several examples showed useless vector control implementation because the target species was not a malaria vector. Okay, so I'm done with malaria. I don't know if you want to have a little pause, if you want a break now, small break, or I can continue speaking about other vector-borne diseases. Up to you.
Yes, I think
Dr. Triwibowo, tell me what you want to do.
yes, I think better when we continue for already your presentation, and then maybe after this we have time for discussion. Maybe it will be better. Yeah.
Okay, so if you are not saturated,
Please, if you need some break, I will give time for you.
No, I'm okay. It's just for
Okay.
you because I speak so much that maybe you are tired now.
Oh, no. It's very interesting. So I think we can continue. Yep.
Okay, thank you.
Okay, you go.
Okay, so I'm going to. So we keep we skip malaria now, and we are going to speak about arboviruses. Oh, not arboviruses only, but vector-borne diseases, because I would like to start with lymphatic filariasis, and that's due to a parasite, so it's not an arbovirus. This is a very crowded slide, but I just would like to show you a few things here. Concentrate on Indonesia, your country, where you have this diversity of lymphatic filariasis, where you can find Bancroftian filariasis in orange. You can find also Brugia malayi. This is Wuchereria bancrofti, the species. Brugia malayi causing Brugian filariasis in yellow. And you have also a small spot here of Brugia timori. And the vector is Anopheles barbirostris. I spoke about it before. So here in Indonesia, you have very diverse situation about lymphatic filariasis. And what is very important to understand is that these different filarial worms are being transmitted by not one mosquito, not two, but 6 genera of mosquitoes. This is really a disease that is can be transmitted by Anopheles mosquitoes, Aedes, Culex, Mansonia, Ochlorotatus, Downsymia. So this is, to my knowledge, the only disease that can be transmitted by so many genera of mosquitoes. So that's the point I want to make about lymphatic filariasis. Diversity in Indonesia and diversity of vectors. Now, for the arboviruses, I think you know what arboviruses means. It's arthropods Vector-Borne viruses. So arboviruses. For these arboviruses, we have the dengue, chikungunya, Zika viruses. Vectors are Aedes aegypti, Aedes albopictus, the tiger mosquito. There is no vaccine, no treatment. And here you have the symptoms for each of them. Although the symptoms look like, we can say that the dengue, you can be very tired for up to 3 weeks. I mean, you know dengue, maybe some of you got dengue, so it's really a painful disease that you can be very tired for some time. Chikungunya, you have severe pain in the articulation, in the arms, legs, muscle pain. And Zika, well, 80% of the Zika cases are asymptomatic, but the main problem is for pregnant women that may have their fetus with microcephaly. So I'm going to speak about dengue. The distribution of Aedes aegypti and dengue cases is shown here in this map. So in blue, the areas infected with Aedes aegypti, and in red, the different places where occur dengue epidemics. And it's important to understand that there are 4 strains of dengue virus 1, 2, 3, and 4, which nowadays are distributed everywhere. They are present worldwide, these 4 strains. So the Aedes mosquitoes are vectors of dengue virus, but not only. They are also vectors of yellow fever, chikungunya, Zika, Eastern equinoencephalitis, and some other encephalitis, as well as filariasis, as I said before. So the dengue is widespread around the world. 4 serotypes, 4 strains, as I said, no vaccine, no treatment, as I said. There is an estimated 100 to 400 million cases per year, among which 96 million cases, clinical cases, and 36,000 deaths. Compared to malaria, it's about 17 less deaths than malaria, just to give you a comparison. So this is annually, of course. And there are more and more cases of hemorrhagic cases of dengue. 70% of the dengue hemorrhagic cases occur in Asia. And so this disease is originally from Asia, but now we have dengue in the Caribbean, Latin America, and more recently, even in Europe. And now we have also cases in France of autochthonous cases of dengue in France. In Africa, there are few epidemics, non-hemorrhagic, on the East Coast and rare on the West Coast, although the 4 serotypes of dengue circulate, and there are, of course, Aedes vectors are present. So this is kind of a mystery why there is no dengue cases, not so many dengue cases among African populations. So the dengue cycle can be sylvatic, so in the forest with some of the vectors here, and also in the cities you can have cycle of dengue in cities with the different vectors listed here. So sylvatic, enzootic cycle, and urban epidemic cycle in cities. This is a very interesting distribution map that I want to show you. It's the evolution of dengue in the within the 70 years. So starting in 1943 until 2013, you can see that in the 70s, 9 countries had reported cases of dengue. So then, yeah, you can see here, it's not easy to, yeah, okay. So the cases are here found in Asia, in 40s, 50s, 60s, 70s. So you can see it's moving into Asia. And then in the 80s, it went to Africa and Latin America. And then it's exploded in Latin America in the 2000. So now we have in 2019 129. Oh, why? sorry. In 2019 there are 129 countries declaring cases of dengue. So you can see that in 20, 30, 50 years, we jumped from 9 countries with dengue to 129 countries. And we jumped also in 2000 from 505,000 cases to 5.2 million cases in 2019. So really an explosion of dengue throughout the world. Now for chikungunya, we have, as I said, chikungunya is known as the curled up man. So chikungunya means curled up man in Makonde language in Tanzania. That's why we call it chikungunya, because the people that get this virus have articulation pain very severe, up to the point that they cannot even hold chopsticks or a fork. You see, the hands cannot hold anything, and they get fever. So it's very problematic disease. So you can see that this disease started in Africa. So the first virus was found in 1952 in Africa, in Tanzania. And then the distribution exploded in Africa and jumped from one continent to the next. And since 2007, there has been outbreaks in the Caribbean, in Latin America, in USA, in Europe. The vectors are Aedes aegypti, Aedes albopictus. And what is very interesting is that in 2005, there has been outbreak, I mean, not an outbreak, a large epidemic in Indian Ocean, and especially in the Réunion Island. Réunion Island is a French island close to Madagascar. And they had no Aedes mosquitoes there. So, and then Aedes albopictus came into the island before 2005. So this mosquito was developing in this island year after year. And in 2005, someone from India came with the chikungunya virus in his blood. And in India, the strain that had this patient had a mutation. The chikungunya virus mutated to be more fit to the transmission by our Aedes albopictus. And then this person, these persons, maybe several, came to Reunion Island. And so the mosquito was there. And this new strain of chikungunya, very well adapted to Aedes albopictus, was found in the Reunion Island, and altogether the mosquito and this new strain generated a very large epidemic in the Reunion Island in 2005 and 6, with hundreds of deaths, and 1/3 of the population got chikungunya between 2005 and 6. So you can see that when the population moves, they bring the virus, the mosquito is there, everything is all together to have these epidemics. So the chikungunya, yes, there are large epidemic in Republic Democratic of Congo. As I said, a very large epidemic in Reunion Island, also in Gabon later on. In 2005 in Indonesia, also large epidemic, reporting with among these countries 1.9 million cases, etc. I mean, outbreaks in the Pacific Island, and then in 2015, more than 1 million cases in Latin America and the Caribbean and USA, with nearly 200 deaths. So for Zika, Zika is a virus that was first identified in monkeys in the Zika forest. That's why we call it Zika, in Uganda in 1948. And in 1952, we recorded the first human cases in Uganda and Tanzania. The main vectors are Aedes furcifer, Aedes africanus, but also Aedes albopictus, Aedes aegypti. And also it was found in Anopheles mosquitoes and Mansonia mosquitoes. Up to now, we don't know if they are vectors, but the virus was found in these mosquitoes. There has been Zika outbreaks in Wap Island in 2007, and from this major outbreak in Yap Island, people came to the American continent, and an epidemic started in 2015 and 16. And you may remember this major outbreak in Brazil and in all Latin America, with numerous cases of microcephaly in children. I think we are nearly the end, so I'm going to speak about some encephalitis. So Culex quinquefasciatus is the responsible for the transmission of several encephalitis, such as St. Louis encephalitis, Lacrosse, West Nile. So the Culex species, I mean, there's not only Culex quinquefasciatus, but other Culex can be vectors of arboviruses, such as West Nile, occurring in Sub-Saharan Africa, Middle East, Mediterranean basin. In Europe, we have West Nile now occurring big time in Europe. The vectors can be different Culex in Africa, different species in Europe, India, USA. St. Louis encephalitis, also occurring in the American continent, is being transmitted by different Culex. Japanese encephalitis, you know this disease occurring in Southeast Asia, transmitted by Culex tritinorhynchus, Culex gelidus vishnui. etc. Or there in Australia also Murray Valley encephalitis. So I'm going to focus a little bit on the West Nile virus, which is a virus transmitted by Culex mosquitoes to birds. So this is the regular cycle. But sometimes a mosquito searching for a blood meal will incidentally bite a human or a horse. This is really incidental, but the problem is that humans and horses can get the disease and may die from it. There has been outbreaks of West Nile in the world, especially in the USA in 1999. Very important outbreaks on the east and west coast, with nearly 2,000 deaths. And I mean, there are many outbreaks of West Nile. I didn't want to show you all the outbreaks, but I want to focus only on the West Nile that we have in Europe. So in Italy, Germany, France, Spain, etc. Every year during the summer, we have outbreaks of West Nile in Europe, affecting horses and humans. And for example, now in this year, 5 European countries reported human cases of West Nile, such as France, Greece, Italy, Serbia, and Spain. And we know that there will be more cases this year. Last, Japanese encephalitis, major cause of viral encephalitis in Asia, with 30,000 to 50,000 new cases per year. The major vector is Culex tritiniorancus and Culex gelidus. The major reservoir are natural hosts such as birds, and the amplification of the virus are porks, and the transmission to humans from these mosquitoes occurs especially in rural areas of Asia. So I'm going to end pretty soon. I just want to show you that the serial killer is really the mosquito. Since 170 million years, we have this serial killer, which is mosquitoes. So we have to protect ourselves. And in conclusion, vector species identification allows the estimation of entomological inoculation rate, pathogen transmission, in the sense of, is it a vector, efficient vector, not a vector? So this is after identification of the species, we can tell, yes, it's a vector, it's not, and to what point it's a good vector. With the species identification, also we can analyze the behavior, the trophic behavior, biting pattern, seasonality of the mosquito. If you have a cocktail of species, you will not know if the mosquito bites better at 6 p.m. or 4 p.m. or whatever, or early in the morning. You have to separate the species. Same for the resistance to insecticide. You have to know what species you have. The bionomics of the vector, its distribution, the ecology also. And in order to implement efficient vector control program, you have to use these techniques, molecular techniques, to target the proper vector species. So I would like to end by showing you the 2 books that I was the editor, and they are in open access. So you go to Intake Open books, you type my name, and you will have access to this book with 24 chapters, free to get the entire book you can click and you can download each chapter. And that's the number 30,092 of the number of the book. And it was published in 2013. And the second book I edited with my colleague from India, Dr. Vasdev, a specialist of malaria in India. This also is in open access. So the number is 6339, but it doesn't matter. You type my name and you will get into the book, and you can download the 17 chapters if you want. And this book has been published in 2018. So really, I thank you so much for your attention, and I'm ready for all your questions.
Okay, thank you very much, Professor Sylvie, for a very excellent and then insightful presentation, of course. So it's very, very interesting during 1 hour and 30 minutes. So I think now we have some time for question and answer. So if you have a question from the participant, please raise your hand. Or maybe if not, you can type in the chat box. So please, maybe is there any participant that have question for this? Please raise your hand, maybe. Okay, if not, I think we still have some question from the chat box. One is for the first, I would like to translate from the ledger pribadi about the where sperm of mosquito sperm is stored in the ovary, and how the transovarial mechanism occur.
Yeah, no, it's stored in very specific organs called spermatheca.
Okay.
The spermatheca holds the spermatozoid of the mosquito, the male mosquito, and when the female is gravid, has eggs, when the female will lay eggs, she will have a spermatozoid in the eggs to have the eggs. How can I say that? Yeah, the eggs formation will need, of course, a spermatozoid. So, but the female will regulate this spermatozoid into her eggs, and she will lay eggs one by one after this, using the spermatozoid that she has herself in the spermatheca. That's why a female mosquitoes usually doesn't mate more than once. Hello? I lost you. Hello? I lost you, Dr. Triwibowo.
Sorry, Professor Sylvie, because I get disconnected for my internet. Okay, yeah, okay. And then for the second question, I think from Purita, I also try to translate. She asked about the how. Could you please to explain the respiratory system in the adult mosquito? The respiratory system.
Yes, the Aedes and Culex, they have a siphon which goes at the surface of the water, and that's the way the respiration of the mosquito will occur. For the Anopheles mosquitoes, the larvae will be parallel, and they have an orifice at the end of the abdomen from which they can breathe. So they don't have siphon, but the end of the abdomen has the proper way to breathe. So that's the last segment of the abdomen is transformed into a respiratory system.
Okay, hopefully it's clear for Burita for the answer from Professor Sylvie, and then maybe because of we have now we have question from the Bu Andi. I would like to try to translate. Number one is, when can we say that a species complex is a new species? And then second one, what is the effect of the existence of species complexes in a controlling vector-borne diseases? And then number three, why is
No,
malaria?
one by one.
Okay, one by one. Okay, sorry, Professor. When can we say that species complex is a new species?
Okay. So, for example, in Indonesia, we know that we have new species like the Java form, or different species of the barberous trees or maculatus. So we know that we have a different species. First of all, and in every study you will do, you will still have to check for the morphology, of course, because you can sort out. And when the morphology is not exactly corresponding to what you find usually, you have to apply the PCR. And then the PCR, if you don't get any bands on the PCR, that means that, well, it's not the species you are supposed to find. So that's a good way to know that maybe you have a different species. So the next step is to amplify a gene that is called ITS2, internal transcribe number 2, that with the universal primers, so that will give you the whole sequence of the ITS2 of the specimens you don't know about. You sequence, you send the, so you do a PCR, you will get a band, you cut the band and you send it abroad, or I don't know, maybe in Indonesia, you have people that can sequence, or maybe in your lab if you are lucky. you do the sequence, you look at the whole sequence of the ITS2, and then you put this sequence into the gene bank, and you look if the sequence match with a species that has already been put into the gene bank. If there is no sequence looking or being close to your sequence, well, that's another proof that maybe you have a new species. So the next step will be to really look at the different stages, the eggs. If you are lucky, you can get the eggs of this population that you don't know. You look at the larvae, the pupa, and then the adults, and you carefully check all the morphological characters. And then you compare with what exists already, and then you describe, if it's a new species, you describe the morphology, you describe you give the ITS2 sequence, and then you can say that it's a new species.
Okay, thank you, Professor Sylvie. And then second question from Bu Andi is, what is the effect of the existence of the species complexes in a controlling vector-borne diseases?
So what I said, species complex, within a species complex, you have vectors, excellent vectors, secondary vectors or local vectors and non-vectors. So when you implement a vector control, you want to target the main malaria vector or the main vector, whatever the disease. And to be efficient, the vector control has to target the right vector. So if you have in an area different species, and it's the case in Asia, because in Asia you may have Aedes mosquitoes, Anopheles mosquitoes in the same place, Culex. So if you want to apply a very efficient vector control, you have to know the biology of your mosquito. Is the mosquito biting during the day? If it's an Aedes, is the mosquito biting at night? At what time, etc. And all these different factors will tell you when and how to implement your vector control. And species identification is part of what you need to know, because as soon as you know what species you have, you can study the biology. And if, for example, the mosquito bites at 6 p.m., you will tell the people to use a repellent or to wear a long sleeve or long pants. If the mosquito, the species bite at 1 in the morning, people are sleeping. So you propose them, or you advise them to use a bed net and sleep under a bed net. So you see, each species will bite at a different time. So you have to know at what time, what is the vector to have a very efficient vector control. So it depends on the mosquito, the time, the behavior, etc. But for an efficient vector control, you really need to have all this information.
Okay. Hopefully Ibu Andi are satisfied with what Professor Sylvie explained. So I think Bu Andi still have many questions, but I think the time is limited. So I think I will move to the Bu Fitriana. She asked about maybe in the chat box. I would like to ask a question. As we know, Indonesia has an abundant population of Aedes albopictus. How significant is the impact of Aedes albopictus in transmitting the dengue virus? Due to Wolbachia program in Aedes aegypti mosquito, where the Wolbachia bacteria have been found in Aedes albopictus, in the future, is there a possibility that Aedes albopictus could become the main vector for dengue like Aedes aegypti? Yeah.
Yeah, absolutely. Aedes albopictus is in France. We don't have Aedes aegypti in France, but in France, Aedes albopictus is a very good dengue vector. When people come with the virus in the blood, the mosquito during the summer, like for example now, it's summertime, the mosquito is there. If people are traveling with the virus in their blood, mosquito will take the virus, and then it's very efficiently transmitting the virus to the people around. So the people that have never traveled around. So we have autochthonous cases now of dengue in Europe, in France. So yes, I say that Aedes aegypti is probably a better vector of dengue virus in Asia, but albopictus, if the Wolbachia program is working well against Aedes aegypti, and so you lower the burden of dengue due to Wolbachia in Indonesia, like Yogyakarta, where you are doing a lot of work. But you have to be careful and check for Aedes albopictus, because this is also very good vector. So you have to find a way to control this mosquito efficiently. So I don't know about the Wolbachia program against Albopictus in Indonesia. I think you are targeting only Aedes aegypti in Indonesia.
Yes.
But
yes.
yeah, some work will have to be done with Albopictus also in the future. I mean, maybe not. I don't know. But today, the work you are doing with Wolbachia on against Aedes aegypti is really a model in the world. You are doing an excellent job using this technique, and you are well advanced compared to other countries. So really, it's excellent job that you are doing against Aedes aegypti. But keep in mind that you have a second good vector, and don't forget about it.
Yes, I agree with you, Professor Sylvie. Maybe we need also pay attention for the Aedes albopictus during the Wolbachia program. It is essential to prevent the potential transmission from the albopictus. Yes, I think thank you very much, Professor Sylvie. Maybe we still have many questions, but I think maybe would you please maybe send the question to us, or maybe by email, maybe so we can send to Professor Sylvie if you still have many questions about it. This is a very interesting lecture, I think, today. So thank you for all of your great question. So maybe before we close this lecture, could you please maybe you have any talk or comment before we wrap up, Professor Sylvie? The final remark before this lecture.
A final remark is that really I enjoyed doing this lecture. I hope it was not too dense, but I am available if you want to contact me. Maybe I will not answer right away because I will be on vacation next week. So, but I will try to answer your questions, and I hope to see many of you at the ICTMM Congress in Kuching, Borneo, Sarawak on September 19 until 23. I will be there. I know that some of you will be there, and the more, the better. And that will be great to have a discussion with you in Kuching during the ICTMM Congress. So please come, and I need to talk to you, and we can have discussion about mosquitoes, and I will be happy to talk to you about that.
Thank you very much, Professor Sylvie. Okay, this is today we have more than 140 participants that join in this lecture. I'm very happy for this, and from various institutions and from various students from many universities. So I think this is, I hope, will give benefit for all of us. So thank you all for your active participation, especially in the chat box, because of I would like to apologize because of not all of the question can be answered because of the time is very limited. So we appreciate your presence today for all participants. And the last, of course, thank you very much for Professor Sylvie Manguin that give the time very nice time for 2 hours. So look forward to seeing you at our next event. So thank you very much again to Professor Sylvie and all of the participants here, and see you and have a wonderful day. Okay, thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Salam, terima kasih Patri, terima kasih Prof.
And
Sylvie.
I hope to continue this collaboration with you and with many Indonesians. I'm open to collaborations, and I'm very proud of the work Dr. Triwibowo has done so far. So yeah, I am with you.
Yes, yes, of course. We still continue our collaboration, strongly collaboration. Yes, thank you very much, and see you.
Bye-bye.
Have a nice day, Professor Sylvie. Bye bye.
Bye-bye. Thank you so
Okay.
much. Bye-bye.
Yeah, bye bye. Yeah. Oke, terima kasih Bapak Ibu. Kuliah pada sore hari ini kita tutup. Assalamualaikum warahmatullahi wabarakatuh. Ya.
Waalaikumsalam warahmatullahi
Waalaikumsalam,
wabarakatuh.
Pak Tri. Terima
Thank
kasih.
you Pak Tri, thank you very much.
Sami-sami.
Terima
Ya.
kasih Pak Tri.
Oke, ya kita tutup bersama perkuliahan sore hari ini. Ya. Terima kasih Pak Tri. Sami-sami.
Terima kasih Bapak Ibu semuanya. Izin kami end nggih.
Dia Mas. Matur
Nggih,
suwun.
matur suwun Pak Tri.
Sami-sami.
Sehat selalu Pak Tri.
Sehat selalu teman-teman. Kemudian
Ya, siapa?
izin di